my goal here is to sort of show you just a little more about just sort of the general kind of system that I'm working on and the kinds of things that it can be useful for. And I'll show you, I guess, a few projects and hopefully try to keep it a little bit light. Let me just jump right in and talk about the kinds of things that I study. The kind of molecules that I study are called Dieblock copolymers. Now, you probably know a polymer is basically any sort of long squiggly chain of a bunch of different pieces stuck together. That's what a polymer is. For instance, the ballpoint pens that you're writing with are probably made out of plastic, and so that's a polymer. That's polystyrene. That means that the individual molecules are lots and lots of styrene blocks that are stuck together into very long chains. The molecules that I work with are actually two different species that are stuck together. So one of them might be, for instance, polystyrene, lots and lots of styrene blocks stuck together. Another one might be something completely different, polyisoprene, polyethylene propylene, something. For the purposes of this talk, we can just think of them as A and B. And all of the polymers that I study, the two sides are a little bit different. You can think of one side as being more or less hydrophobic, and one side is being sort of more or less hydrophilic. So think of them as sort of like oil and water. And of course, you know what happens with oil and water, or like oil and vinegar in a salad dressing. Or you shake it up, and of course it starts to spread apart with the oil on top. If you think about it, what these molecules are like is if you could separate all of the B parts, or sort of the B chains from the A chains, and mix them together, they'd separate like a salad dressing. But notice that in this case, the two species are actually linked together as part of the same molecule. And that means that they can't separate macroscopically, like the salad dressing does. But it's still true that if you're an A part of the molecule, you want to hang out with other A parts, and if you're a B part, you want to hang out with other B parts. So these molecules do the best they can, and what they end up forming is actually periodic assemblies that look, for instance, like this. So here I have this is sort of a three-dimensional block of this material, and it actually is made up of layers or lamellae, where each sort of the blue part here is the blue ends of a bunch of molecules that have all been stuck together, and they're sort of all randomly kind of twisted around like spaghetti in there. And what's nice and what's interesting about this is it really is in fact a periodic pattern, where the periodicity, the lattice constant, is not equal to the length of the molecule, but it's related to the length of the molecule. Just to give you a sense of scale here, this is always fun. Atoms, of course, are very tiny, sort of on the scale of angstroms. Human hair and other sort of macroscopic biological systems are much, much larger on the scale of microns. Visible light is also sort of handfuls of nanometer, hundreds of nanometers. Typical spacings for these systems, it depends on the size of the molecule chain, but typically tens of nanometers is about the size that we're talking here. So the key here is really, really honkin' small is what these things form. Now, here are a couple things to keep in mind. There are a couple of other neat things that these molecules can do. Let's imagine that you could sort of go in here and sort of snip off the ends of all of the blue molecules and make them shorter. And let's think about how this structure would change. Well, of course, the obvious thing would be that, of course, the blue parts would start to get smaller, and if you made the red parts longer, of course, then the red part would expand. But to some extent, if you just simply made the blue parts of the molecule shorter, the blue parts here would just sort of get thinner. Okay. Here's what happens, though. That happens for a point, and then at some point, these things get so thin that they actually sort of break off into individual cylinders of blue material. And again, the structure of this is that each of these blue cylinders in here is actually sort of the blue ends of the molecules that are all blue side in on this cylinder. So that cylinder is actually made up of sort of the conglomeration of lots and lots of different molecules. And you can play this game a little more and say, gee, what happens if I again go in and if I could sort of magically go in and make the blue ends of these molecules even shorter? Well, for starters, the cylinders would start to get smaller and smaller and smaller, and then at some point they'd break up again into little spheres. And so this is actually kind of a neat trick. Actually, this is something that should be familiar to you. Have you ever seen, for instance, rain dripping down a window pane? And you think about what happens when you have a lot of water flow. What happens is you get this sort of nice steady stream, and then if the water flow gets sort of smaller, the stream gets thinner and thinner and thinner, and then at some point it breaks up into individual drops. Essentially, it's the same physics that's governing this. What's happening is because the blue parts don't like to hang out next to the red parts, we can think of there as being some sort of energy cost associated with this boundary. And so it's simply figuring out the point at which cylinders would break up into spheres is simply a matter of saying, well, which geometry is actually the one that's going to minimize the surface area between these two species. So a bunch of different things that these materials can do. I'm basically going to talk about only spheres for the rest of this talk, though. And in fact, the things that I'm going to talk about are all very thin layers of these spheres. I've shown you sort of pictures of sort of these three-dimensional blocks of this stuff. In fact, I'm going to, for most of my work, I end up putting thin layers of this on a surface. So I have a hard surface, like a silicon wafer. And I simply put a very, very thin layer of this material on the surface just to form a single layer of spheres. So this is a layer that's on the order of about 25, 30 nanometers thick. And looking down on the top of this, you would actually see a regular lattice of spheres arranged on the surface. Again, each sphere is actually the conglomeration of a few dozen or a few hundred individual molecules kind of wadded up together in a little ball. The other kind of system that I'll show you is if we make a coating like this on a substrate that's not completely flat, but actually has a little step edge carved in it, it turns out that we're actually able to line up these balls with the step edge. Essentially, the very first layer of spheres wants to stay a certain distance away from that step edge, and then the second row of spheres tends to sort of line up in the notches between the first row, and this order sort of ends up percolating out this way then from that. And that actually is a very useful technique if in fact what you want is a well-ordered rows of spheres. And this leads sort of one of the most general problems that I look at in my lab with my students is how does order form in these materials? When I first make a film of this material, it sort of forms these little spheres, and you can see this is a microscope picture of these spheres. Sure, they're spheres, they're kind of disordered. They're not all the same size, and they're certainly not very well arranged. On the other hand, at a later time, which is to say after I've elevated the temperature just a little bit and allowed individual molecules to kind of shimmy around and get comfortable, so to speak, it actually does end up forming a pretty good hexagonal lattice. And you can see in this picture to the right that these spheres are in fact all aligned pretty nicely in rows in a hexagonal lattice, and each one of these spheres has sort of 6 nearest neighbors all around it. So, I mean, this is actually a really interesting question in physics and, to some extent, in mathematics and materials science as well. How does this order form? How do you go, really, from this disordered state to this ordered state? And it even gets a little more complicated. You can see that maybe there are sort of regions here where all of the rows are aligned very well, and then maybe there's another region where the rows are aligned in a slightly different direction. At some intermediate stage, you get sort of lots and lots of these little grains, these little sort of regions where the crystal orientation is in a particular direction. And then over time, some of these little grains melt away and others of these grains get larger. So how do you go to forming a sort of coarse pattern of sort of large grains? This is sort of related to, well, it's a universal question in material science, in fact. On the lower picture, what I have here is, this is a picture of a substrate that actually has one of these step edges in it. So this is the step edge here, and the low side is to the left, and the high side is to the right. And see, again, initially when I first cast the film, it looks kind of jumbled up and it's a wreck. But in fact, at later times, lo and behold, these spheres have actually formed nice little rows here. And so sort of seeing how you get from one to the other is sort of the central question that I'm trying to answer in my lab. Incidentally, a word about the pictures that you're seeing here. I guess the top one, I think, was with a scanning electron microscope. That's a little older one. This one, however, is with a microscope that I have in my lab called an atomic force microscope. And you should really take a minute to appreciate the scale here. Each one of these little spheres in one of these pictures is something like 10 nanometers across. There's something like 20 nanometers spacing between two of these spheres. Which means that the entire picture that you're looking at here is something like 1 micron across, maybe just a little more than 1 micron. So keep in mind, a human hair is 50 microns, so a human hair on this scale is sort of from here to sort of way past that wall someplace. So it really gives you an appreciation of sort of the power of this microscopy. Let me tell you a little bit about how this microscope actually works. This is what we actually have up in my physics lab on the 2nd floor here at Gottwald. It's a neat microscope. The way it works actually is there is in fact a really sharp tip, which is basically it's a single crystal of silicon. Actually, the entire top part here is carved out of a single crystal of silicon, and it's oriented so that sort of the sharp point of the crystal is aimed right down at the sample. And so the way this microscope works, believe it or not, is by physically approaching the surface and simply and finding where the surface is, and then physically scanning back and forth and feeling what's underneath it. And again, think about the engineering that's required to do that, to actually position something to within sort of a nanometer or two, and then raster it back and forth. Now the other thing that this microscope does is it can actually tap on the surface and actually feel how hard the surface is beneath it. So for instance, in the polymers that I study, if the short part of the molecule is polystyrene, that's kind of a hard plastic like your ballpoint pens, the part in between the spheres is actually polyisoprene or polyethylene propylene, which are kind of softer, rubbery, they're kind of snotty polymers, really. So when this tip sort of goes back and forth and taps, imagine it's sort of, there's a bowl of Jell-O with a bunch of little grapes sort of buried in it, so the tip is sort of tapping and sort of feeling where the grapes are underneath. And it's sort of amazing that it's able to do this. Here's a picture of the microscope actually in our lab. These are two of my students actually who just graduated. This is Nate Lawrence and Jill Kehoe. Nate's now an engineering student at Boston University. This is the microscope in the background and the computer that's controlling it and some other electronics we have hanging out. It's all enclosed in an acoustic shield hood, floating on a cushion. So it's able to take these really nice pictures. So turns out though, taking nice pictures only gets you so far in this game. That if you really want to understand things, you have to do things quantitatively, and you have to actually analyze your images. And people always forget this. If you have a pretty picture, that's still only data. And it's really easy, especially for our mind, to be fooled by a pretty picture into thinking that we're seeing what we want to see. So we always take a lot of care to analyze our images quantitatively. And let me give you an idea of how we do that. Here's an image of a bunch of spheres. One of the things we do is actually use computer software. Rather than sit around with a pencil and say, here's one sphere, here's another sphere, well, I guess there's another one here. We actually use a computer program that we've written, software that we've coded ourselves, to figure out sort of a good representation of the center of each one of these spheres. So we can find that. I mentioned that these are in fact lined up in rows in a hexagonal lattice, and sure enough, there's an analysis we can do to pretty much figure out mathematically what the nearest neighbor is for each one of these. And we see that in fact this is the same data represented here on the right, where I haven't shown the initial picture, but I have shown simply the connections from each sphere to its sort of local neighbors. And you see that each one sits in the middle of a hexagonal cell. And here again, it's very easy to see from the lines that these are lined up in rows. Next thing to do is look at each individual unit cell and say, gee, okay, if this cell here has a particular orientation, the one next to it has about the same orientation, this one over here has kind of a different orientation. Now just to help us see that, I'm going to apply a sort of color map to this, where depending on sort of the orientation, the local orientation of each individual unit cell, I'm coloring that group of pixels with a color along this wheel. And looking at this, it's very easy to see that yes, pretty much the sort of the lower left-hand side of this image is in one orientation, and sort of the upper right is another orientation. And there's actually a grain boundary that runs through the middle, and I can then analyze things about that grain boundary. And that's how I can really analyze quantitatively sort of the size of the region over which there's a sort of a well-defined orientation. I wanted to show you an actual movie that shows how this kind of order develops in time. So check this out. This is an atomic force microscope picture. You may be able to just see little spheres in the back, and I've overlaid this color information, where the color corresponds to the local orientation of that hexagonal crystal. And again, there's this step edge here in this film, and one of the things, and as I step time forward, and so what I'm doing is I'm sort of allowing these molecules to wiggle around a little bit, and then I take another picture, they'll let them wiggle around a little bit and take another picture. And as I do that, we see a couple things. First of all, we see that over the course of time, we do go from lots and lots of little random grains to a few larger grains. But the other big picture here is, look at this big green area. The green color in this film is what corresponds to orientation, as it turns out, along with that step edge. So you see that this orientation with the step edge is just sort of working its way further out, and eventually it'll sort of take over the whole screen, kind of like kudzu or something. So studying this kind of thing quantitatively is the kind of thing that we do in my lab. Who cares? There are two reasons why people care about this. The first reason is, well, it's cool, it's neat. I mean, if you want to understand how the world works and how crystals form, I mean, this is a very general problem, and it's applicable not just to Matt's little crystals made of polymers, but pretty much any other crystal you can think of. Metals, gemstones you dig up out of the ground, pretty much anything. Understanding it is nice, but actually doing useful stuff with it is even nicer. And it turns out that people are really interested in these materials as sort of a way to make really, really tiny things as part of nanotechnology. So let's see, IBM and Intel and AMD are all making computer chips. They all try to cram lots and lots of little transistors onto a chip. Anyone know what a typical size of a transistor is for Intel or AMD? Handfuls of nanometers, actually. It's close to about 100 nanometers, actually. That's about the smallest transistor that they can make. So about 100 nanometers. Wouldn't it be nice if we could make something that was even smaller, say 10 nanometers or 15 nanometers? Gee, about the size of the little spheres and cylinders that are made out of these Dieblock copolymers that have self-assembled into these nice patterns. Okay, maybe you want to do that, but these polymers aren't computers, right? I mean, they're not made of silicon, they don't conduct electricity. I mean, from a computing point of view, they're really boring. But the nice thing about them is you can use the fact that they self-assemble into these patterns, and then use that pattern as sort of a template for building other things. Let me show you, here's one example of how you might do that. For instance, if the polymer that you choose is something like, for instance, a polybutadiene species, which my chemist friends tell me has a double bond in it, which oxygen likes to attack and make go bye-bye, then in fact, if you bubble this thing in an aqueous solution with some extra oxygen in it, it'll essentially get rid of these spheres or cylinders and leave you with voids. And then you can go in and process these voids with, for instance, a plasma etch that sort of etches down into the material and leaves then this nice sort of surface that has polymer left in some places, but not in other places. And here's the key. Once you get to that stage, it turns out you can make anything that you want. For instance, here are a couple different ways to go. If you already have this, one thing you can do is sort of coat the surface with, for instance, a metal. And this could be a magnetic material or a conducting material or a semiconductor or something interesting like that. And then use a solvent to sort of float away, dissolve away the polymer, and float away the metal where it doesn't actually stick in the surface, leaving you with now metal patterned on the surface where the polymer used to be. And it turns out that all of these steps here are relatively easy once you can get the top step. So once you can get the top step, you're golden. Here's another way to go about this, is if you knew ahead of time what you wanted and you coat the whole thing with metal, then you can do essentially the reverse process, using an acid or something to etch away the metal in some places, using the polymer as sort of a mask, protecting the metal in other places. Again, get rid of the polymer and be left with metal in some places but not others. And this is essentially the kind of processing steps that people use in the microfabrication industry for computer chips and stuff like that. Let me give you, of course, you do have to worry about, well, gee, how do I make these things form the shape of a transistor or the shape of a graphics card, a graphics processor or floating point unit or something like that. Well, obviously that's hard to do. But as a first step, let's suppose that you just wanted to make something for storage. You just want to store bits of information. So let's think about how a hard disk works in your computer. A hard disk is basically a glass platter coated with a magnetic material, and it spins around and around, and the read-write head positions itself over it and essentially changes the magnetic field locally over a portion of that disk. So for instance, if you look at a cross-section of the surface, different areas of that magnetic material on the surface might have the local magnetic field pointing up or down. This one might be up, and the next one might be up or down. And these represent logical ones and zeros, which are, when you store a photo or store a message or something. Then this is, of course, how the information gets stored. It turns out, though, people are always trying to cram more stuff onto a hard drive, make bigger and bigger hard drives, but make them physically smaller. And one of the problems they run into is when you cram these bits close together, if they're pointing, the magnetic fields tend to flip each other. You actually get a little crosstalk between the magnetic domains, which if you think about it is really bad for data storage, right? You really don't want that to happen, for the bits to suddenly spontaneously flip, right? Bad idea. So it turns out there's a nice way to cheat and manage to cram these magnetic domains closer and closer. And that is, if instead of having a uniform magnetic coating over this surface, if you can actually manage to microfabricate a whole bunch of itty-bitty, teeny-tiny magnetic posts with a non-magnetic material around them, then it turns out you can actually cram these bits closer together than you otherwise could, and they won't be able to have this crosstalk and flip each other. So this is a possible way to basically cramming more information onto the same surface area of a disk. Here's another one. This is a possible new transistor made by Intel. It has lots and lots of, although it's only one transistor, it has sort of lots and lots of little ridges on it. And again, I look at this, from my point of view in ICG, a lot of pseudo-periodic ridges, all spaced about 20 nanometers apart. Hey, I bet I can think of a way to make that nice and cheap. This, by the way, is a picture of the clean room at VCU, Virginia Commonwealth University. And I actually worked with a guy in the electrical engineering department using some of his fabrication facilities to make various devices for experiments related to this kind of stuff. And it's hard to tell who's who, of course, under the little bunny suits, but my guess is that that's Gary Atkinson showing other people how to use. I guess that's a furnace of some kind. Let me just tell you, so this is sort of the broad picture of the kind of stuff that I'm working on. Let me just go very quickly into a few specific actual projects that I'm working on. One is, so I've talked about using this nice, this really nice microscope for doing imaging. It turns out that we actually have some pretty good control over it, and we're hoping that we can actually use it also to manipulate samples. I mean, as long as we have this tip that can position itself to plus or minus a nanometer, let's see if we can go in there, maybe flatten the tip a little bit, and actually try scooching it around on the surface and use that to try to align some of these spheres. People have actually tried this alignment and been successful with it before, but only on a much larger scale, sort of centimeters at a time. And what I'd like to do for the first time is try to go in and try to align only only a few rows of these spheres at one time. And then I could actually do controlled experiments for particular grain boundaries and actually design an experiment as opposed to simply starting from disorder and going to order randomly. One of my students, David Hoffman, helped make a new kind of device for for actually coating this polymer onto a surface. It's actually a really nice setup. It's computer controlled and allows us, for instance, to make a film which is thin at one end and then thick at another end in sort of a gradient, which is nice if we actually want to study something as a function of film thickness. And Sarah Atwater, a senior right there, is actually just starting to work with me on this project as well. Incidentally, on the first project, Brent Fallon started to work with me. He's now working with Ted, and Cosmin Pankratov, who's now in computer science, both worked for a while on this project. Nate Lawrence and Jill Kehoe, who you saw pictured before, have been working on aligning these spheres with step edges. There's plenty more work to do there. There are a bunch of variations in that experiment I'd like to try out. Actually, skipping down a little bit, Cosman and Jake have both worked on, again, in all of our data analysis, we end up doing a lot of math and a lot of basically writing a lot of code and doing computer science. And it turns out that we stumbled on a really nice way to make an algorithm very, very fast. And we wrote it up. And so these two were actually co-authors on a series of 3 journal articles that just got published. The last thing I want to talk about is this project with Dan Katz, who's at this point, he's long gone, and Brian Salmons, who just graduated last year, did this really nice project which has just gotten some really nice results. There's this thing that happens when you take an atomic force microscope picture. It turns out that it's very hard to get a picture that's really exact and precise and isn't at least a little bit distorted. And the problem is this. Fundamentally, you have to image by resting, by holding this tip above the surface and scanning back and forth. And so you really have to position this very carefully to within about a nanometer. And the problem is that positioning can drift over time. For instance, as a function of temperature. If the microscope heats up just a little bit and the sample expands or contracts a little bit, then suddenly you can be imaging a slightly different part of it. So for instance, if you were imaging something that looked like a checkerboard, it might end up looking a little bit skewed or a little bit stretched out in one dimension, just basically because the image is moving under you as you're sort of slowly scanning this tip back and forth over it. And what I'm showing you here is actually toggling back and forth between 2 different images of the same area. Now you can see I've centered this, this one little L shape. This is a very disordered polymer sample. This is in the same place in both pictures, and you can tell the area around it is sort of moving a little bit to the left and the right as I toggle between the first and the second image. Nate Lawrence actually took these 2 images, just one right after another, and it just shows how each image is just a little distorted. So what Brian Salmons was able to do with some code that he wrote is look at each image individually. So I mean, separated the 2 images from each other, and worked to correct the distortion in the first image, and also worked to correct the distortion in the second image. And then when we brought them back together, here's the corrected versions. So you see that the first image actually did get sort of shifted over to the right a little bit. And the second image I'm overlaying on top. And it's even hard to tell, other than these sort of margins where it's changing, it's actually hard to tell what's actually going on. But in fact, this is that same feature that we were looking at, and everything else is pretty much staying in the same place. So we've reduced the amount of distortion in these images to about 1 pixel. And that's actually a really happy result for us, hence the happy face there. Kathy asked me to talk specifically about what it's actually like to work in my lab. So I tried to figure out what it would be like, because I mean, you do get to do a lot of different things. I mean, there's a little bit of mixing up samples together and actually coating them. There's playing with kick-ass expensive microscopes. Oh, incidentally, I say microscopes because not only is the atomic force microscope, but we've also done some work with Carolyn Marks in the Biological Imaging Center using one of her microscopes as well, a laser scanning confocal microscope that's really a gonzo machine. So we've used that as well. We do write a lot of our own computer code for doing image and data analysis, and we also end up trying to extend what our microscope can do by sort of adding extra electronics and instrumentation on it and basically trying to beef it up. Occasionally we get to visit the VCU clean room, or I also collaborate with folks at the National Institute of Standards and Technology, so we get to go up there and see what they're up to. And I have brought students to conferences. For instance, I had a student give a talk in LA, and I had a student give a presentation in Denver. Jill went to Denver with me. We didn't actually make it to New Orleans. And of course, then you have to put up with me. And let me just close by saying I'm actually looking for more students to work with. I had a really, one of the nice things about teaching here is you get to see your students go off and do great and wonderful things. Unfortunately, last year, all of my students went off and did great, wonderful things all at the same time, and they all graduated, and now I have no one working for me anymore. Come back, please. So anyway, I really am eager to find some new students to work for, and if you're interested, come and see me.